Welcome to Bookfulness. This is episode 2 of our deep dive into evolution, the 4th edition by Shutuma and Kirkpatrick.
Yay there.
So last time, we really set the scene, didn't we? We talked about the history, Darwin's big ideas, descent with modification, natural selection, kind of the groundwork.
Exactly. We established that evolution happened.
Yeah.
But how did it happen? What path did it take? That's what today is all about.
Right. So today we're diving into Chapter 2, the Tree of Life. This is all about the evidence for common ancestry, how scientists map out this huge family history.
Yeah, it's the roadmap. Phylogeny, phylogenetic trees, that's the language we use to trace how everything is connected.
Our mission then for this deep dive is to really unpack that. How do they build these trees? How do you even read one? And what do they tell us? Because some of the relationships they reveal are, well, pretty surprising.
Definitely counterintuitive sometimes. We're moving from the theory to the actual traceable history. Okay, so the chapter kicks off with this really foundational idea, the unity of life. If everything evolved from a common ancestor, then deep down, all life should share some fundamental operating system.
Like we're all running slightly different versions of the same software.
That's a great way to put it. And the evidence isn't, hidden away. It's right there in our genes. The book starts with a killer example, the eyeless gene experiment.
Ah, yes. I found this one fascinating. Tell us about it.
Okay, so fruit flies have a gene called eyeless that's crucial for, well, eye development. Obvious name, right?
Makes sense.
And vertebrates, like us, or mice, have a functionally equivalent gene called PAX6. Now, the amazing thing is how similar these genes are in their job, despite flies and mice being separated by hundreds of millions of years of evolution.
Hundreds of millions. So their last common ancestor was tiny and ancient, way before dinosaurs, right?
Way before. We're talking deep, deep time. And the book explains why this gene, PAX6 or eyeless, is so conserved. It's a master switch.
A master switch.
Yeah, it's a transcription factor. It doesn't build the eye proteins directly. Instead, it tells a whole cascade of other genes, okay, team, build an eye here.
Ah, I see. So it coordinates everything.
Exactly. And because it's coordinating so much, any big mutation in that master switch gene would just mess everything up downstream. Development would likely fail catastrophically.
So natural selection keeps it really stable, locked in almost.
Pretty much locked in, which makes the experiment they did just, well, mind-blowing.
What did they do?
They took the mouse version, PAX6, and they put it into a fruit fly embryo. And not just anywhere, but they triggered it in weird places, like on the fly's leg or its antenna.
Okay, so mouse gene in a fly. What happened? Did it make a weird little mouse eye?
That's the incredible part. Now, the mouse gene, working in the fly cells, successfully ordered the fly's genetic machinery to build a fly eye, a compound eye, the correct type for a fly, just, growing on its leg.
Oh, hold on. The mouse gene built a fly eye.
Yes, you can see it in figure 2.1 again. If you have the book or the notes, check out the figures. It shows that the mouse gene understood the fly's instructions, used the fly's building blocks, and followed the fly's blueprint.
That could only happen if the underlying language, the commands that PAX6 gives, are fundamentally the same in both animals, right? Conserved all that time.
Precisely. It's astonishing proof of that shared operating system, that deep common ancestry.
And it wasn't just this one example, was it? The book mentioned another one.
Right. They reinforce it with a yeast and human gene swap experiment. Yeast and humans. We're talking maybe 1 billion years divergence there, even further back.
1 billion years. Okay, so what did they swap?
They looked at about 414 genes in yeast that are absolutely essential for survival. Like, the yeast just dies without them. Basic life support stuff. Then they replaced those essential yeast genes one by one with the corresponding human gene, the human ortholeg.
And did the human genes work in yeast?
Nearly half of them did. Almost 200 human genes could perfectly substitute for their yeast counterpart and keep the yeast alive.
That's amazing. So for almost half of these critical survival functions, the fundamental genetic code is interchangeable between a fungus and a mammal after 1 billion years.
It really hammers home that unity. It's the bedrock upon which the whole idea of the tree of life rests.
Which brings us, directly to Darwin's big metaphor that all life branches out from common ancestors like one giant family tree. You can see representations in figures 2.3 and 1.5b from the last chapter.
Yep. But if we're going to map this tree properly, we need some rules. We need clear definitions for the groups we're identifying on the tree. This is where taxonomy gets really specific. Box 2A talks about this.
Okay, taxonomy. Why is it so important to get the definitions right? Like, if we know birds came from dinosaurs, why does it matter if we call the old reptilia group one thing or another?
It matters hugely. Because if your group definition doesn't accurately reflect evolutionary history, it obscures the real relationships. It leads to bad science, wrong predictions. Modern biology aims only for what are called monophyletic groups, or clades.
Monophyletic. Okay. What's that?
A monophyletic group, or a clade, includes a common ancestor and all of its descendants. Think of it like a branch you can snip off the tree cleanly, taking everything that grew from that point. Mammals are a perfect example, one ancestor and all mammals descended from it.
Okay. Ancestor plus all descendants. Got it.
Okay.
What are the other kinds, the ones we avoid?
Well, the next one is a paraphyletic group. This includes the common ancestor, but, and this is key, it leaves out some of the descendants, usually for like historical or superficial reasons.
Ah, like the traditional reptiles.
Exactly. The old class Reptilia included turtles, lizards, snakes, crocodiles, but it left out birds.
Even though we now know birds evolved from dinosaurs, which are nested right within that reptile lineage.
Right. So by leaving birds out, Reptilia becomes paraphyletic. It doesn't represent a complete branch of the tree. It suggests falsely that crocodiles are somehow more closely related to lizards than they are to birds, which isn't true based on their shared ancestry. It messes up the history.
Okay, so paraphyletic is bad because it's incomplete. What's the third type?
The third type is polyphyletic. This is, well, it's usually a real mess. It's a group where the members do not share an immediate common ancestor within that group. They're lumped together often just because they look alike or live similarly.
Based on superficial similarity, not real history. Any examples?
The book gives the old example of falconiforms. Traditionally, this group included falcons and hawks because, well, they're both birds of prey, right? Hooked beaks, talons.
Yeah, they look the part.
But the DNA evidence came along and showed falcons are actually more closely related to parrots and songbirds. They just convergently evolved that hunter lifestyle and look. Lumping falcons and hawks together was polyphyletic, an artificial group based on appearance, not ancestry.
Wow. Okay, so we need monophyletic groups, clades, to build an accurate map.
Precisely. And once we use those proper groupings, the phylogenetic tree becomes this incredible history book. Figure 2.5 maps some major events onto the tree.
What kind of events jump out?
Well, you see really deep history, like the origin of mitochondria. That was endosymbiosis, an ancient cell basically engulfing a bacterium, which then became the powerhouse organelle.
A bacterium inside our ancestor cells. Wild. What else?
Then there's that relationship that always gets people, animals and fungi.
Right, I read that. We're closer to mushrooms than to trees.
Evolutionarily speaking, yes. The phylogeny shows animals and fungi are sister groups. We share a more recent common ancestor with each other than either of us does with plants. Things like having chitin, a structural molecule, point to this shared history. It totally reframes how you think about multicellular life.
It really does. Any more recent but still surprising connections?
Oh, yeah. Birds and crocodilians.
Alligators and, say, sparrows?
They are each other's closest living relatives. Their last common ancestor lived over 200 million years ago, back in the Triassic. But they are still closer to each other than an alligator is to a lizard, for example.
So when you see them together in the Everglades?
You're seeing two branches from the same relatively recent part of the reptile. Well, the archosaur part of the tree. It completely blows up any classification based just on what things look like today.
Okay, so we have this map built on monophyletic groups, showing real ancestry. Now, how do we actually read it properly? The book mentions two types of change shown on these trees.
Right. First, there's cladogenesis. That's the branching process itself. Speciation. When one lineage splits into two or more new lineages.
Like when the lineage leading to humans and the lineage leading to chimps split from their common ancestor. That split is cladogenesis.
Exactly. That's what creates the tree structure, the diversity. The second type of change is anagenesis.
Anagenesis was that?
That's the change happening within a single lineage along one of those branches over time. Even if a lineage doesn't split, it's still evolving, accumulating changes.
So, like, if you follow the lineage from the first feathered dinosaurs all the way to modern birds, the gradual changes in feathers, flight, beaks, that's anagenesis.
Precisely. It's the transformation along a branch, while cladogenesis is the splitting of branches. Both are happening constantly.
Okay, so to understand these branching patterns, we need the right vocabulary. The book diagrams this in figure 2.6. Let's break down the parts of a tree.
Sure. The lines themselves are the lineages or branches, showing the path through time.
Right.
Where two lineages split apart, that point is an internal node. That node represents the most recent common ancestor of everything branching off from it.
Okay, the ancestor is the node.
The whole diagram usually starts from the root, which is the inferred oldest common ancestor for all the species shown on that particular tree.
The base ancestor.
And we already defined a clade that's everything descended from a single node, a monophyletic group. And finally, the two clades, or species that split from the same immediate node, are called sister groups.
Like humans and chimps bonobos are sister groups.
Exactly. They're each other's closest relatives on that tree.
This brings us to something you mentioned earlier, the biggest mistake people make. Looking at how similar things look instead of how recently they share an ancestor.
Yes, this is absolutely critical. Evolutionary relationship is only about the recency of common ancestry. It's about the nodes, the branching pattern. How similar things look today can be incredibly misleading.
The book uses that shark and dolphin example, right? Figure 2.7.
Perfect example. Sharks and dolphins look superficially similar, streamlined body, fins, predators, but phylogenetically, a dolphin is a mammal. Its closest relatives are things like hippos.
Much, much closer than to a shark, which is a fish.
Way closer. The common ancestor of dolphins and hippos lived far more recently than the common ancestor of mammals and cartilaginous fish. Looks can deceive. The tree reveals the history.
Okay, so don't judge by looks. What's the other common mistake in reading trees?
It's reading meaning into the order along the top or side. People often assume that species listed, say left to right, are somehow progressing from primitive to advanced.
Ah, like a ladder of progress.
Exactly, which is completely wrong. This is where the rotation principle is important, figure 2.8.
Rotation principle. What does that mean?
It means you can rotate the branches around any node, like spinning a mobile, and the relationships don't change.
So if a tree shows A, B, C, D along the top, B and C might share a recent node. You could flip that node so it reads A, C, B, D, and it means the exact same thing about who is related to whom.
Precisely. The only thing that matters is the connections which branches connect at which nodes. The order they happen to be drawn in linearly is just, well, an artifact of drawing a tree on a flat page.
So no species is more evolved than another just because it's on the right side of the diagram.
Absolutely not. Think about it. Time runs from the root past to the tips present. All living species at the tips have been evolving for the exact same amount of time since their lineage split off from the others depicted. There's no higher or lower among living things in that sense.
Okay, we know how to read a tree, we know the pitfalls, but how do scientists actually build one? Where does the data come from?
It comes from comparing homologous characters. Homologous means the features are similar because they were inherited from a common ancestor. These shared features are the evidence.
Like the bones in our arm, a bat's wing, and a whale's flipper. Same basic bones, modified for different jobs, showing shared ancestry. That's homology.
That's the classic example, the pentadactyl limb. Another amazing one the book mentions is the tiny bones in our middle ear, the malleus and inventus.
Yeah, what about them?
They are homologous to bones that formed the jaw joint in our reptile-like ancestors. They got repurposed for hearing in mammals. You can trace the transition in the fossil record and development. It's shown in figure 2.9.
Wow. Repurposed jaw bones in our ears. But today, it's mostly DNA, right? How does DNA work as a character?
Yeah, DNA data revolutionized phylogenetics. Basically, each position in a DNA sequence is treated as a character.
Like position number 57 in a particular gene.
Exactly. And the character state is simply which nucleotide base A, T, C, or G is found at that position in each species you're comparing.
So you end up with massive tables of species versus DNA bases at thousands of positions.
Huge data sets. Now, the problem is, with even a modest number of species, say 20, the number of possible trees connecting them is astronomical, just unbelievably huge.
So how do you pick the right one? Or the best hypothesis, anyway?
You need a logical principle. The most common one, especially historically, is parsimony.
Parsimony. Sounds like being cheap or simple.
That's exactly the idea. The principle of parsimony says the simplest explanation is usually the best. In phylogeny, this means we prefer the tree that requires the fewest evolutionary changes, the fewest mutations, gains, or losses to explain the character data we see.
The tree with the least amount of evolutionary stuff happening on it.
You got it. Minimum number of steps.
How does that work with DNA? Let's use that squirrel example from the book, feature 2.10, to make it concrete.
Okay, so the goal is to figure out the relationship between the three tree squirrels: fox squirrel, eastern gray, and western gray. We also have a ground squirrel as an outgroup.
The outgroup helps figure out what the ancestral state was, right?
Correct. So let's look at just one DNA site, say site 3 in their example. The outgroup, ground squirrel, has a T nucleotide. The fox squirrel also has a T.
Okay.
But both the eastern gray and the western gray squirrels have an A at this site. So T is likely ancestral, and A is the derived state shared by the two gray squirrels.
Right. So we have three possible ways to connect these three tree scores.
Yep. Hypothesis 1. Eastern gray and western gray are sister species, closest relatives.
Okay. If that's the tree, how many changes happened at site 3?
Well, if they're sisters, their common ancestor must have had the T to A mutation. That single mutation then got passed down to both of them. So that tree requires only one evolutionary change, T to A, to explain the data at site 3.
One step. Okay. What about hypothesis 2? Fox squirrel is sister to eastern gray.
Now it gets messy. Fox has T, eastern gray has A, and the other one, western gray, also has A. To get this pattern on this tree, the T to A mutation would have had to happen independently twice. Once in the lineage leading to eastern gray, and again separately in the lineage leading to western gray.
That's 2 steps, 2 separate mutations.
2 steps. And hypothesis 3, where fox is sister to western gray, has the same problem. It also requires 2 independent T to A mutations to explain why both eastern and western grays have A.
So tree 1 needs 1 step. Trees 2 and 3 need 2 steps each.
Exactly. Therefore, parsimony tells us to prefer tree 1. It's the simplest explanation. It minimizes the required evolutionary changes for that site.
And scientists do this for thousands of DNA sites.
Thousands, even millions now. They calculate the total number of steps required by each possible tree across the entire dataset. The tree with the overall lowest score, the most parsimonious tree, is considered the best hypothesis for the relationships.
It's not absolute proof, but it's the strongest statistical inference based on minimizing the evolutionary changes needed.
That's the logic of parsimony, yeah.
Okay, so parsimony helps find the most likely branching pattern. But is evolution always a neat, clean branching tree, the book says? Not always.
Right. Life is messier than that sometimes. Sometimes branches that diverged can actually merge back together. This is called reticulate evolution. It makes the history look more like a net or web in places.
How can branches merge?
One major way, especially in plants but also sometimes in animals, is hybrid speciation, figure 2.11.
Hybrid speciation, like 2 different species making a new third species.
Exactly. Imagine species X and species Y hybridize. Often their offspring Z are sterile, like mules. But sometimes, maybe through doubling of chromosomes or other genetic events, the hybrid offspring Z can become fertile and reproductively isolated from both parent species X and Y.
So Z is a brand new species formed by the joining of X and Y. That definitely makes a loop in the tree.
A literal loop. Another way things get tangled, especially in the microbial world, is horizontal gene transfer, or HGT, sometimes called lateral gene transfer.
Horizontal, meaning not parent to offspring.
Correct. This is genes moving sideways between organisms that aren't directly related through reproduction. Bacteria do this, constantly swapping genes for things like antibiotic resistance. That's a huge deal in medicine.
How do they swap genes like that?
Through a few mechanisms, picking up naked DNA from the environment, transformation, getting genes transferred by viruses, transsection, or direct cell-to-cell transfer, conjugation.
Does HGT happen in more complex organisms too?
It does, though maybe less frequently than in bacteria. The book gives a really stunning example in aphids, figure 2.12.
Aphids, what genes did they get horizontally?
Genes for making carotenoids. Carotenoids are pigments, like the ones that make carrots orange. Animals normally can't make them. We have to get them from our diet, typically from plants or algae.
Okay. So aphids can make their own.
Some can. And when scientists looked at the genes responsible for making carotenoids in these aphids and built a phylogenetic tree just for those specific genes.
What did they find? Where did the genes come from?
The aphid carotenoid genes didn't group with other insect genes at all. They fell smack bang in the middle of the fungi lineage on the gene tree.
They got pigment genes from a fungus.
The evidence strongly points that way. An ancestor of these aphids somehow acquired the entire metabolic pathway for making carotenoids horizontally from a fungus. It allows them to produce their own colors.
That's wild, a gene-jumping kingdoms. So HGT really can scramble the history, making parts of the tree look more like a web.
Definitely for bacteria, their deep history is probably more web-like than tree-like because HGT is so common. It makes tracing their evolution really challenging.
This leads to another complication, doesn't it? The difference between the history of a species and the history of a single gene within that species.
Ah, yes, the species tree versus the gene tree, or gene and genealogy. They are not always the same. Figure 2.3.
How can they be different?
It's often due to something called incomplete lineage sorting, or ILS. Imagine an ancestral species that has two different versions or alleles of a particular gene, say allele A and allele B.
Okay, variation in the ancestor.
Now, that ancestral species splits into two new species, species 1 and species 2. Initially, it's likely that both new species will inherit both alleles A and B, just because the split was recent.
So the gene versions were already sorted, in a way, before this species fully split.
Sort of, or rather, the sorting isn't complete at the time of splitting. The ancestral variation persists across the species boundary for a while. Then, maybe species 1 splits again into 1A and 1B. Just by random chance, allele A might get fixed in 1A, and allele B might get fixed in 1B, and maybe species 2 kept allele A.
Okay.
If you then build a tree based only on that gene, it might look like species 1B and species 2 are sisters because they both have allele A. Maybe allele B was lost in species 2 later, even if the true species history is that 1A and 1B are the actual sisters.
Wow, okay. So one gene's history can give you the wrong species relationship if you're unlucky with how the ancestral variation sorted out.
Exactly. That's why modern phylogenetics relies on using data from many, many genes, thousands of them. The idea is that the true species signal will emerge as the dominant pattern, averaging out the noise from individual gene trees affected by ILS.
Makes sense. Use more data to drown out the conflicting signals. Now, there's another layer of gene history complexity related to gene duplication, right? Orthologs and paralogs.
Yes. Gene duplication is incredibly important. It's a major source of evolutionary novelty. When a gene gets accidentally copied, one copy can carry on the original job, leaving the second copy free to mutate and potentially evolve a brand new function.
Like having a backup allows you to tinker with the spare.
A perfect analogy. This process of duplication creates two distinct types of homologous relationships between genes, figure 2.14.
Okay, what are they?
First, you have orthologous genes, or ortholegals. These are homologous genes in different species that diverged because of the speciation event. The mouse PAX6 and the fly eyeless genes are orthologs. Their history reflects the history of the mouse and fly lineages splitting.
Okay, orthologous trace species splits. What's the other type?
Paralogous genes, or paralog. These are homologous genes that exist within the same species or lineage because they arose from a gene duplication event.
So duplication makes paralogs. Speciation makes orthologs. Can you give an example of paralogs?
The classic example is the globin gene family in humans, figure 2.15. We have myoglobin in muscles and various hemoglobin genes, alpha, beta, etc. in blood. They all carry oxygen. They're clearly related, homologous.
Right.
But the gene that became myoglobin and the ancestor of the hemoglobin genes didn't diverge when humans split from chimps. They diverged because of a gene duplication event way, way back in an early vertebrate ancestor, maybe 600, 800 million years ago.
Ah, so myoglobin and hemoglobin within humans are paralogs.
Exactly. They arose from that ancient duplication. Now, the human beta hemoglobin gene and the chimpanzee beta hemoglobin gene, those are orthologs. They diverged when humans and chimps speciated maybe 6 to 7 million years ago.
I see. So the paralog divergence, myoglobin versus hemoglobin ancestor, is ancient, reflecting the duplication event. The ortholog divergence, human beta versus chimp beta, is much more recent, reflecting the speciation event.
Precisely. And you can see why telling them apart is critical. If you mistakenly compared a human paralog with a chimp ortholog, thinking they were both orthologs, you'd calculate a ridiculously ancient divergence time for humans and chimps. You have to compare apples to apples ortholog to reconstruct the species tree accurately.
Okay, we've navigated the complexities of building and interpreting these trees. Now let's talk more about what we can do with them. You said they're like history books. How do we read the story of a specific trait?
We use a technique called phylogenetic mapping, or ancestral state reconstruction. You take your reliable species tree and then you map onto it the presence or absence of the trait you're interested in. Maybe it's feathers or warm-bloodedness or a specific behavior.
So you color in the branches where the trait appears.
Essentially, yes. And by looking at the pattern, often using parsimony again, you can infer where and when the trait most likely evolved or was lost. Let's use the hominoid toe example from the book, figure 2.16.
Okay, our toes versus chimpanzee toes.
Right. We walk bipedally, so we need our big toe aligned with the others for push-off. It's non-opposable. Chimps, bonobos, gorillas, orangutans, they retain the ancestral condition of an opposable big toe, useful for grasping branches.
Like a thumb on their foot.
Pretty much. So we map this trait, non-opposable big toe, onto the primate phylogeny. We see it only on the human branch.
So when did it evolve?
The mapping shows it must have evolved after our lineage split from the chimpanzee bonobo lineage, but before the diversification of the genus Homo. It pinpoints the evolution of this key bipedal adaptation to a specific segment of our evolutionary history.
It turns bipedalism evolved into bipedalism evolved here on this part of the branch. Much more precise.
Exactly. It lets you sequence events. Did bipedalism come before big brains or after? Mapping traits helps answer those questions.
Can we get actual dates from these trees, not just relative order?
Sometimes, yes, using the concept of the molecular clock.
The molecular clock? Sounds intriguing. How does it work?
The basic idea proposed back in the 60s is that mutations, especially in certain parts of the DNA, might accumulate at a relatively constant average rate over long periods, like a clock ticking away evolutionary time.
So more genetic differences between two species means more time has passed since they split.
That's the core concept. If the rate is steady, the number of DNA sequence differences is proportional to the divergence time.
How do you know the rate? How do you set the clock?
You need to calibrate it. You find lineages where you have reliable divergence dates from other sources, usually the fossil record. These are your calibration points. Figure 2.17 shows this.
Okay, like fossils show species A and B split 50 million years ago.
Right. So you sequence DNA from A and B, count the differences, say there are 100 differences. You then find another pair, C and D, known to have split 25 million years ago, and maybe they have 50 differences. You plot these points differences versus time.
And hope they form a straight line.
Ideally, yes. The slope of that line gives you the tick rate of the clock for that gene or that group of organisms, e.g., 2 differences per million years.
And once you have that rate,
You can apply it to lineages without good fossils. If you find 2 other related species, E and F, and they have 80 DNA differences, you can estimate using your calibrated rate, 2 decimer, that they must have diverged about 40 million years ago.
so it lets you date splits even without fossils. That's powerful.
Hugely powerful, especially for things that don't fossilize well, like bacteria, viruses, or many soft-bodied organisms. It's how we estimate the timing of deep evolutionary events or track recent viral outbreaks.
But the book adds a caveat, right? This clock isn't always perfectly steady.
Definitely not. That's a major limitation. The rate of molecular evolution isn't universal. It varies between different genes. Some evolve fast, some slow. It also varies significantly between different groups of organisms.
How so?
For instance, organisms with faster metabolic rates or shorter generation times, like mice, tend to accumulate mutations faster than slow metabolism. Long-generation organisms like primates or turtles. DNA repair efficiency also plays a role.
So you can't just borrow a clock rate from mice and apply it to humans.
Absolutely not. You need to calibrate the clock specifically for the group and the genes you are studying. It's more like a collection of molecular stopwatches than one universal clock. But used carefully, it's an invaluable tool.
Okay, so we've built the tree, read it, dated it. Looking at the whole tree of life, what are the big overarching patterns that emerge? The book highlights a few key ones.
The most fundamental one goes back to something we touched on earlier. Evolution is a tinkerer, not an inventor. It works by modification of pre-existing features.
Things don't just pop into existence from nowhere.
Never. New structures are always modifications of something the ancestor already had. The raw material for novelty is the ancestral anatomy, physiology, or genetics.
Like the mammal ear bones from reptile jawbones.
Or wings in birds, bats, pterosaurs, all modified forelimbs, using the same basic bone structure inherited from their non-flying ancestors, just stretched, fused, or lightened in different ways. Evolution is constrained by history.
This tinkering, this modification, leads to another pattern, doesn't it? Mosaic evolution.
Yes. Mosaic evolution is the principle that different characteristics within a single lineage evolve at different rates. An organism isn't evolving uniformly in all its parts.
So one feature might change rapidly, while another stays almost the same for millions of years.
Exactly. Which means that any living species is a mixture, a mosaic, of traits inherited from ancient ancestors, plesiomorphic traits, and traits that evolved more recently within its own lineage, apomorphic or derived traits.
And this is why it's scientifically wrong to call one living species more advanced or more primitive than another.
Precisely. That whole ladder of progress idea is busted by mosaic evolution. Take a frog. Its lineage branched off before mammals did, so you might think primitive. But frogs have highly derived traits, like the loss of teeth in the lower jaw. In many species, that's an apomorphy, a new thing. Yet they might retain the ancestral 5 toes on their hind limb, a plesiomorphy.
And humans. We have the ancestral 5 fingers, plesiomorphy, but a derived single bone in our lower jaw, apomorphy, unlike our reptile ancestors.
Right. Every species is a patchwork of old and new. Since old versus new depends entirely on which trait you look at, ranking organisms on a single scale of advancement is meaningless. They're all just successful survivors in the present day, each with its own unique mosaic of traits.
Okay, so modification and mosaicism. What's the last big pattern?
Homoplasy. This is when similar traits evolve independently in different lineages. It's the opposite of homology, similarity due to common ancestry. Homoplasy is similarity without recent common ancestry possessing the trait. The main type we talk about is convergent evolution.
Conversions, different starting points, same destination basically, driven by similar environmental pressures.
Often, yes, similar lifestyles favor similar adaptations. Think of the streamlined shapes of dolphins, mammals, and ichthyosaurs, extinct marine reptiles, and sharks, fish, all converged on that efficient swimming shape independently.
The book uses a really striking example of convergence, the vertebrate eye and the octopus eye, figure 2.20.
Ugh. Ah, the eyes. It's a classic. On the surface, they look remarkably similar, don't they? Both have a lens, an iris, a retina. It looks like a perfect design, maybe even evidence for design to some.
Yeah, superficially they seem almost identical.
But look at the wiring. That's where the independent, constrained history reveals itself. In our vertebrate eye, the light-sensing cells, the photoreceptors, point backwards, away from the incoming light.
Backwards?
Yeah.
That seems weird.
It is a bit weird, and because they point backwards, the nerve fibers, axons that carry the signal, have to run across the inner surface of the retina, gather together, and then punch through the retina to form the optic nerve leading to the brain.
And that punch-through point, that's the blind spot, isn't it?
That's our inescapable blind spot, a design flaw inherited from the way our ancestors' eye happened to develop. Now compare that to the octopus eye.
How is it different?
It evolved completely independently from a different tissue origin. And in the octopus eye, the photoreceptors point forwards towards the light, like you'd expect. The nerve fibers come out the back of the photoreceptors and exit the eye cleanly behind the retina.
So, no blind spot.
No blind spot. It's, in that sense, a more sensibly wired eye. It achieved the same function image formation, but through a different evolutionary path, unburdened by our specific ancestral wiring constraint.
Wow. The fact that the octopus eye is better wired, while ours has an inherited flaw, is actually really powerful evidence for evolution by modification, not perfect design.
It's one of the strongest arguments, I think. Evolution doesn't aim for perfection. It tinkers with what's already there, flaws and all. That suboptimal design in our eye is a clear signature of its history.
And the book wraps up this section, Box 2B, by saying all this comparative evidence, homology, suboptimal design, the shared genetics we started with, would point to evolution even if we didn't have a single fossil.
That's the argument. The patterns in living organisms mapped onto phylogenies tell a consistent story of descent with modification from common ancestors. The fossils are fantastic confirmation, but the core evidence is arguably right here in the comparative biology of life today.
Okay, before we finish, let's just quickly nail down that parsimony logic again with the simple problem from the end of the chapter, just to make sure we've got it.
Good idea. It's fundamental. So the setup is we have DNA from 3 hypothetical island species, species 1, 2, and 3, and we have their mainland ancestor, species 4, as the outgroup.
Right. We look at one specific DNA site. The outgroup 4 and species 3 both have a G, but species 1 and species 2 both share a unique mutation, an A, at that site.
Okay. G is ancestral, A is derived and shared by 1 and 2. Using parsimony, what's the most likely relationship between 1, 2, and 3?
We want the tree with the fewest evolutionary steps.
Exactly.
Well, if species 1 and 2 are sister groups, then the G to A mutation only needed to happen once in their shared ancestor. Then they both inherited the A. That's one step.
Okay, one step for the 1 and 2 are sisters tree. What if, say, 1 and 3 were sisters?
1 has A, 3 has G, and 2 also has A. To get that pattern, A must have evolved independently in the lineage leading to 1, and again independently in the lineage leading to 2. That's 2 G to A mutations, 2 steps.
Right. And the third possibility, 2 and 3 being sisters, would also require 2 independent mutations to explain the A in both 1 and 2.
So 1 step versus 2 steps. Parsimony picks the 1-step solution.
Absolutely. The most parsimonious explanation is that the shared derived character reflects a shared history. Therefore, species 1 and 2 are each other's closest relatives. That single mutation happened once in their common ancestor.
Simple logic, but you scale that up over thousands of characters, and it becomes a powerful tool for building the whole tree.
That's how it works. Hashtag tag outro.
So wrapping up this deep dive, we've seen how the tree of life isn't just a nice metaphor. It's a scientific hypothesis built using rigorous methods like parsimony, based on concrete data like DNA sequences and homologous structures.
Yeah, we've gone from the profound unity shown by things like that mouse gene building a fly eye, all the way to the nuts and bolts of reading tree diagrams, nodes, clades, sister groups, and understanding why relationships depend on common ancestry, not just similarity.
We also tackled the messy bits, how branches can tangle through hybridization or horizontal gene transfer, and why a gene's history isn't always the same as the species' history.
And we saw the big patterns revealed by the tree, evolution as modification, the mosaic nature of all organisms, and the fascinating phenomenon of convergence, like that octopus eye, all driving home that idea that no living species is more advanced than another.
Next time, we're going to take this map, the tree of life, and focus on the engine that drives the change along its branches. We'll be diving into Chapter 3, Natural Selection and the Evolution of Adaptations.
Looking at the mechanisms that shape life on the tree should be great.
And for a final thought to leave you with, picture that octopus eye versus our eye again. The octopus got the better wiring independently. We're stuck with a blind spot, a legacy of our particular evolutionary path. It's maybe the most striking reminder we discussed today that evolution is a historical process, constrained by the past, building on what came before, imperfections and all, not a march towards perfection.
Couldn't have said it better myself.
Thanks for listening to Bookfulness. Join us for the next episode.